Hello everyone, you all guys are listening me on Unacademy online platform. As you all know very well, it is a biology series for all examinations. And myself is Ravi Agrahari and you people are listening me on Unacademy online platform. This is my lesson number 3 and here I'll discuss with you about the cytology as a cell biology. Cell biology is very lengthy topic, so here I brought in various parts. This is the part number 1. So myself is Ravi Agrahari and I'm a director of Purvanchal IAS at Gorakhpur, where I'm providing you the face-to-face classes. I've written 2 books on McGraw-Hill. The one is on science and tech and second is the environment. This is a highly recommended book for all of you. This is my Telegram channel where you can find out all the entire PDF. So kindly connect with me on my Unacademy profile is unacademy.com/Ravi P Agrahari 20. Over here you can find out my all the courses and the live sessions on Unacademy. My code is Ravi UPSC. During joining any courses, if you people will use it, you will get extra 10% discounts in your fee structure. India's best educator of UPSC CSE, they are taking the classes on Unacademy. It's a great opportunity to all of you to connect with all the experienced faculty for UPSC CSE. Unacademy is giving you CSE as your option if you people are going to take a 2-year subscriptions or more than that. The extra subscription in the case of your requirement, Unacademy is ready to help you with the code of Ravi UPSC. Kindly join it. Even in the case of mains preparations, UPSC CSE test series Unacademy brought for all of you. For general studies and optional both, kindly join with Ravi UPSC code. UPSC CSE Notes Unacademy is 2.0 is providing you with 20 comprehensive UPSC CSE GS booklets with all plus and iconic subscriptions where everything is covered now. Kindly subscribe. UPSC CSE Ultra Combat is on 17 July 2022 at 11 am. Here in 60 minutes you must have to give a test with the 50 questions, a lot of vouchers, gift vouchers. You have options to get it. So finally we are here into the discussion of cell biology. See guys, even in the school level when you have started your studies, I'm very sure you got the first chapter as a cell. Why first chapter as a cell? So here have some reason guys, because cell as it is described, it is a unit of life. Cell is a unit of life and the study of cell, you know, it is called as a cytology. So what is the cytology? Cytology is the study of cell. Okay, and see one thing more here I'll tell you. In the case of cytology, if you differentiate, cytology means cell and logy means study. So overall the study of cell is called as a cell biology. You know why cell is was the first chapter in your school time? The reason is cell is an unit of life. Okay, how cell is a unit of life? So you can see guys, like in the first day, we all people form inside our mother body in the form of zygote. Okay, as like suppose this is the father, suppose like this is a father, and second side is the mother guys. Second side is the mother. During the reproduction period from the father's side, there is a million of the sperm produced, but from the mother side have only one functional egg. So ultimately, what about the option here? Only one sperm fused with the one egg and form the one zygote. So first day we all people was a single zygote structure, you know, and zygote was a one cell structure. So here I can tell you in the first day we all people were the single cell, it was a zygote. You know what happened inside the mother body? This zygote, those form in our mother body guys. This zygote as a single cell structure start to divide and redivide. It form a tissues. This tissue grow and form an organ. This tissue slowly gradually grow and form an organ. This organ has started their own function that is called organ system. This is called as an organ system. Just like whenever your kidney forms, so excretory system start. When your heart came, you know, the circulatory system start. Okay, when your lungs came, respiratory system start. So all the organs slowly gradually they start their own function as an organ system, and all the organ systems have a coordination and form a life. So ultimately a life is what? As a 3 kilogram of baby. Suppose you people born with a 3 kilogram of baby. When you people born with a 3 kilogram of baby, you are a multicellular body. I hope you can understand guys. You are a multicellular of organisms. Okay, and this multicellular of the body even developed from the single cell structure, and that is the reason guys I can tell you, as an organism, those you people born after 9 months and 6-7 days. 9 months and 6-7 days is your gestation period. In this maximum duration of time, we all people develop into our mother body. So a single cell was the reason behind the formation of a life. So cell is a unit of life. Okay, and that is the reason guys, cell is the first chapter also here. To describe the cell guys, 2 German scientists has proposed, even given the cell theory. Okay, the cell theory was formulated by 2 German scientists. The one is a Sliden and second one is a Swan. Sliden was a botanist and the Swan was a geologist, and they have given this cell theory in 1839. Okay, how to define the cell according to cell theory? As I told you guys, cell is a basic structural and function unit of an organism. As here you can see, cell is a basic and structural function unit of an organism. This life is what? This life born as an organism. So whatever is written here in the case of cell theory, I just proved here through the help of this flow chart. Overall, if you go and see all the entire organisms, so they have a different types of cells. Two main cells that you can find out into the all organisms, either it's a plant, animal, everywhere you will see these two specific type of cells. Okay, the most common and popular type of cell is called prokaryote cell, and the second one that you can see in the next screen is called as a eukaryote cell. Okay, the prokaryotes and eukaryote cell are what? I'll tell you one thing, guys. Prokaryotic cells are very primitive cells. They are very primitive cells, and they have a very basic type of animal, like very small type, mycoplasma, bacteria, blue green alga. You know, blue green alga is also called as a cyanobacteria. This is also popularly called as a cyanobacteria. In these organisms, you know what happened? The prokaryotic cell presents. So prokaryotic cells are present actually in the primitive organisms, very basic organisms, very small size of organisms, and they are not very well mature and developed. They lack the nuclear membrane. They have not the nucleus or definite nucleus. Okay, so ultimately in the prokaryotic cell have a less incipient nucleus, a less developed nucleus. Overall, I can tell you as nucleus absent. Okay, another one, guys, in the case of prokaryote cell, the various cell organelles like mitochondria, chloroplast, Golgi bodies, endoplasmic reticulum, these all absent here. In the case of prokaryotic cell, one of the very specific protein that is called histone protein. Histone protein generally not available in the prokaryote, it is totally absent. Okay, but in the prokaryotes have a double stranded DNA, as we have a DNA but not have a histone proteins. Okay, and what is the histone protein? I will explain with you inside the chromosome structure. And this is the reason here it is called as a genophore. Genophore means only DNA not having a histone proteins. So this is the reason in the case of prokaryotic cell, genophore is the correct term. Okay, reason for the chromosome structure, histone is important. Even whenever I'll discuss with you in the coming session, I'll tell you chromosomes have a what? It has a sum of DNA and histone protein. DNA and histone protein. DNA and histone protein. Okay, everyone. But histone protein absent in the prokaryote cell. And if DNAs are present, then it is not chromosome, then it is called as a genophore. I hope you people will remember. It's a prokaryotic cell. So prokaryotic cell is a very basic. Here you will not get the presence of nucleus, less developed nucleus, mostly cell organelles absent, histone protein absent. Example, mycoplasma bacteria and blue green algae. Another side, if you go into the eukaryote cell, they have a proper definite nucleus, guys. They have a proper definite nucleus, well matured nucleus, they have nuclear membrane, well developed nucleolus, everything here, guys. And another one I will tell you, eukaryote have a histone protein, and that is the reason chromosome is the correct term here. But in the case of prokaryote, the correct term was genophore. Reason histone protein was absent. And I spoken you guys, chromosome have a DNA and histone protein, both. Even if you go into the eukaryotic cell have a many cell organelles. What kind of cell organelle? You can see like they have mitochondria, they have plastids, they have endoplasmic reticulum, they have a Golgi bodies, even all. Here you can find out. What about the feature of mitochondria? What about the feature of plastids? What about the feature of endoplasmic reticulum? What about the feature of Golgi bodies? Everyone I will explain you. Wait. And see, what about the example of eukaryote cells? So here I'll tell you guys all the higher organisms like plants and animals. I can tell you guys, we all people are the example of eukaryotic cells. We all people are the example of eukaryote cells. Okay, so eukaryotes are well developed, well mature cells. Everybody remember. Here I just drawn a picture. I just try to show you guys about the comparison between the prokaryotes and eukaryotes. So comparison between the prokaryotes and eukaryotes you can see very easily on the screen. Okay, the well matured, the structure you will see in the case of eukaryotes. Less developed cell you can see in the case of prokaryotes. Okay, here you will see like prokaryotic cell referred to as a simple and unicellular. Definitely, mostly they are simple and unicellular. They lack a nucleus, you all people know very well. Further, it is a repetitions. The nucleoid of prokaryotic cell have only DNA. Okay, and it may be a small and circular any type, maybe double stranded and all. Prokaryotic cell have a less cell parts compared to eukaryote like DNA, ribosome, cell membrane, cell wall. There are other cell parts, they are not present in the prokaryote cell. Mostly you will not get it. An example of prokaryotes you people remember, that is the bacteria, archaeas and all. Eukaryotes example, eukaryotes they refer to the complex and multicellular organisms. As you know, plants and animals, they are complex and multicellular body. They have a nucleus, well developed nucleus and all. DNA or genetic material of a eukaryote cell is found within a cell nucleus. Plasma membrane, cytoplasm, nucleus, ribosome, mitochondria, peroxisome. These all types of vacuoles make up the eukaryote cell as very specific. Okay, and the example you know very well, guys. The animal and plants are the main example of eukaryote cells. Okay, here you can see like picture the figures of prokaryotes and eukaryotes. You can see through the help of this photographs. Finally, I'll come into the discussion of, as you people have seen the cell of two types. You know, cell is of two types. The one you people have seen as a prokaryote cell, and second you guys have seen as a eukaryote cells. You know, prokaryotes and eukaryotes have a many differences. But if I ask you what about many striking difference between the prokaryotes and eukaryotes, so you guys always remember the presence and absence of nucleus. If I ask you what about the main striking difference between the prokaryotes and eukaryotes, you remember there is a nucleus. There are many differences you people have seen, but the main striking difference is what is presence and absence of the nucleus. Everybody remember, that is important part. And here in the eukaryote cell, you people have seen have a presence of plant cell and animal cell. So here I brought a difference between what about the difference between the plant cell and animal cell. But you must be clear, guys, plant cell and animal cell both are the example of eukaryote cells. So one side you see the plant cell and second side you see this is the animal cell. Even the comparison of plant cell and animal cell, you people know very well, it was a UPSC prelims question in 2020. So in UPSC prelims 2020, UPSC asked the question between the comparison of plant cell and animal cells. If you see the plant cell and animal cell, guys, plant cell have an outer layer and this outer layer is called cell wall. Okay, but the cell wall as an outer layer into the cell is absent, is not possessed in the case of animal cell. And this is the main difference in the case in between the plant and animal cell also. So plant cell have cell wall, but animal cell have no cell wall. That is the main difference. Okay, below the cell wall, if you see, guys, okay, there is a presence of cell membrane. Below the cell wall have a presence of cell membranes. That is another layer. And here cell wall is not there, so that's why the outer layer is the cell membrane layers. Here outer layer is the cell membrane. Here outer layer is a cell membrane. You must be clear. Here outer layer is what? Here outer layer is a cell membrane. Even whenever UPSC asked a question in 2020 prelims examination, UPSC asked this one as an option. Second one, plant cell have a chloroplast. Okay, but animal cell do not possess a chloroplast. I'll tell you guys, chloroplast is a cell organelle that is responsible for the photosynthesis. Plant are almost rectangular in the shape. Okay, animals are mostly round in the shape. Okay, that is not very common, but that is also some of the informations. Plant cells possess a large vacuoles. Animal cells have a many and small vacuole. That was the second option that UPSC has in 2020. Plant have generally one vacuoles, rarely two vacuoles. So very less number of vacuoles and they are large in size. But animal cell have many vacuoles and that's why they are small in size. Okay, plant cell have a cilia and flagella type of structures. That's why the plant cell can be moved very easily. But this is the cilia and flagella type of things are absent in the plant cell. But mostly in cilia's type of things, mostly you will get in the case of animal cells. Even centrioles, higher plants do not possess the centrioles. Centriole is also one of the cell organelle that is responsible for the formation of spindle fiber, and a spindle fiber is required for the during the cell division, guys. Okay, but centriole type of feature is not much important. But I'll tell you guys, plant cell do not have centrioles, but animal have a formation, have a centriole as a cell organelles. Plant cell have no or less number of lysosome. Lysosome is a very important cell organelle. Sometimes you people know very well, you people will see the question of lysosome. In the abnormal condition, it is called as a suicidal bag. That I will bring in the coming session. Just wait about the feature of lysosome. Animal cell possesses the many number of lysosome. That's wrong. You just do the correction. It is a lysosome. So here the less number of lysosome, here we'll get the more number of lysosome. Okay everyone, fine. This is the difference of the plant cell and animal cell. You can see in this picture, guys. In the case inside the plant cell, you can see the outer layer cell wall. Okay, here you will see the outer layer cell wall. Just beneath have a cell membrane. But the cell wall you will not see in this case of animal cell. Here the outer layer is a cell membrane. Okay, rest of all feature you will see. Plastid, if you want to see, guys, plastid. The plastid or chloroplast you will see in the plant cell, but it is not present here in the animal cells. So that is the rest of all you will see. It is common. The rest of all you will see it is a common. Okay, it is common. So I hope you can understand very well, guys. Okay, in this case of the plants and animal cell, what about the differences? But again I repeat, what about many striking difference between the plants and animal cell? So you remember that is the presence and absence of the cell wall. See, some of the factual data that I brought for all of you, this may be important for everyone. Okay, if I ask you in this living world, in this entire living world, guys, which one is the smallest cell? So smallest cell you will find into the one of the organism that is called Mycoplasma laddelii. Mycoplasma laddelii is that microorganism which is the smallest one in the living world. So there is a single cell and there is a smallest cell. Even I can ask you one more question, which is the smallest organism in the living world? Still your answer is same, Mycoplasma laddelii. Mycoplasma laddelii is also popularly called as a PPLO. What is PPLO? Pleuropneumonia-like organism. Okay, pleuropneumonia-like organism. Actually, you know, the shape and structure of everything of the Mycoplasma laddelii look like all form of bacteria. Okay, they are looking like they are changing their shape and structure and something like pleuropneumonia-like organisms. That is, and see, this is also popularly called as a joker of plant kingdom. So remember, PPLO is also called as a joker of plant kingdom. As I told you guys, they are changing their own shape and structure every moment of time. In another side, if I ask you which one is the largest cell, largest means area wise. So you remember the ostrich egg is the single cell and it is the largest one. Okay, as a diameter you can see 117 to 155 millimeter. It's a very large in size, it's very huge in size. But one more thing I'll ask you about, which one is the longest cell? Do you know which one is the longest cell? Largest means area and longest means length especially. If anywhere you will see the longest cell, where it is available, so you remember neuron cell is the longest cell that even present inside your body also. So neuron cell is the longest cell. So what about the smallest cell? Mycoplasma laddelii. Mycoplasma laddelii have a many other alternative name. Those people know very well. And largest cell is an egg of ostrich. Everybody know this. Now finally we'll come into the cell structure. Okay, if you go into the cell structure, within the cell you people have seen many of the components. Many components you people have seen inside the cell structure. What about these many components? All the detail I brought here for everyone. See guys, the first I'll start with the cell wall. As you people know very well, cell wall you people have seen inside the plant cells. And see what is the feature of plant cell based cell wall. It provide a definite it have a definite and the rigid cell wall. Plants have a definite and rigid cell wall. As you know, animal cell have cell wall absent. And see guys, I told you guys the difference of many striking difference between the plant and animal cell. You guys know very well presence and absence of cell wall in the animal cells. By nature, if you see about the cell wall guys, it is dead and permeable. It is dead and permeable. Okay, what is the meaning of dead and permeable? So I'll tell you guys. Dead means it has a no need of energy as an ATP and all. Okay, it has a no need of energy as a ATP and all. And permeable means any of the substances may come directly through cell wall across either inside or outside. Okay everyone, what is the feature of cell wall? I'll tell you guys. Cell wall provide a proper shape to the cell. Cell wall provide a proper shape to the cell. The one side it will provide a proper shape to the cell. And second side about, you know, it also protect the internal structure. It also protect the internal structure of a cell. It also protect the internal structure of the cell. Also protect the internal structure of the cell means internal structure means, I hope you can understand very well guys. Okay, the very cell organelles, mitochondria, Golgi bodies, a lot of things are there. Okay, it protect them. The another one guys, when you people will see the cell wall have a different sublayers. Cell wall have a different sublayers. Primary layer, secondary layer, tertiary layer, quaternary layer. It is depend on the various plants body. Somewhere have a less thickness are required. So single layer, primary layer. More thickness are required, more thickness, more rigidity, more activities. So secondary layer also there, tertiary layer, quaternary layer. But one thing I will tell you guys, just like within a plant body, suppose this is one cell wall, between two cell wall have one of the cementing layer. That's why one cell connect with the second cell. So this is the cementing layer. This cementing layer is actually called as a middle lamella. So middle lamella is what? Middle lamella is actually one of the layer of the cell wall, that is a cementing layer. This is cementing layer between two cell wall. So one cell wall connect with the second cell wall with the third cell wall, third, fourth, fifth, sixth. This is what? This is the cementing layer. The bead in between the central layer is called as a middle lamella. Okay, you know what happened? This middle lamella is made up of pectic substances. What about the pectic substances? They are made up by calcium pectate and magnesium pectate. They are made up of calcium pectate and magnesium pectate. You know what is going to be happen during the ripening process? During the ripening process, this middle lamella is going to dissolve. And whenever the middle lamella is going to dissolve, this fruit will become soft. So I hope you can understand guys what happened during the ripening of the fruit. I hope you will remember during the ripening process of the fruit, guys, the middle lamella that is made up by calcium pectate or magnesium pectate, in this period of time into the plant body, there is a formation of pectinase enzyme. The formation of pectinase enzyme into the fruit, guys, they will do the what breakdown, dissolution of the calcium pectate and magnesium pectate, as which is in the case of middle lamella. Now middle lamella dissolve and the fruit will become soft. Okay, so pectiolytic pectinase enzyme, they are responsible to do the breakdown of the pectate layer, and finally this fruit becomes soft. So I hope the cell wall based middle lamella, it may be a good question in your paper and you people will remember. So ripening of fruit is a dissolution of middle lamella. Overall, this is the conclusion. If overall you people will go under the function of cell wall, you already know very well, is the exoskeleton of the cell, provide the mechanical structure to the strength to the cell, a part of provide a definite shape and movement of material in and out of the cell easily through the help of cell wall. The reason I told you cell wall is dead in nature, and because of so very simple, guys, suppose I am dead in nature in my classroom. So any of the student can come directly and go outside without asking me. So I have no value, I have no role inside the class. Okay, so the same type of feature you can see in the case of cell wall. If cell wall is dead in nature, so of course it is totally permeable. Anything can come inside or anything can go outside easily from the cell. After the cell wall, guys, the second one is the plasmid. One more information I will tell you that is very, very important. If you people will go into the chemical composition of cell wall. So cell wall is generally made up by cellulose. So mostly cell wall have a cellulose, and cellulose is a kind of carbohydrates. Even if you people are eating the green vegetables, so what is your food? This carbohydrate as a cellulose is the food. Even other plant have also some other material apart of cellulose, like there is inside the cell wall have possibility of hemicellulose, lignin, pectin. So these all possible, guys. So cellulose, hemicellulose, lignin, pectin, these all are possible. Okay, everyone. But one thing more, guys, mostly cellulose, and cellulose is very, very common. You always remember. Okay, you always. Even one thing more I will tell you, those lignin is the chemical composition if it is more in the cell wall. So that's why the cell wall become very, very hard. So if I ask you the strength of cell wall is what, the simple answer is what? More amount of lignin content into the cell wall provide more and more strength. Do you know the paper pulp industry? In the case of paper pulp industry, paper manufacturing industry, we people are using the plant-based material, and those plant-based material inside the cell wall have a less amount of lignin content. That cell wall based, that plant-based cell wall will be used for the development of better quality based paper. Okay, so more amount of lignin create the problem in the paper pulp industry. So more amount of lignin, paper quality is worst. Less amount of lignin, better paper quality. So in that way, you people should know in this reference of cell wall. So lignin is in the reference of paper quality, that one questions, and middle lamella as in the case of fruit ripening is the another questions. So these are the good question for all of you. And if anybody are eating the green vegetables, so what is your food? Cellulose. So you people are going to digest the cellulose as a carbohydrate. Okay, cell membrane. Now we are here into the second cell organelle, that is called cell membrane. Cell membrane, or you can call as a plasma membrane. Cell membrane, you know, it is the inner layer into the plant cell and outer layer in the animal cell. Everybody know. It is living because it has a need of energy, and that's why it is living, so it is selectively permeable. It will check the material and then allow. Either anything can come inside, it can go outside. Inside the cell membrane, guys, if you see, within the cell membrane have a protein components around 52%. Within the cell membrane, so maximum inside the cell membrane have a protein. The second one is a lipid constituents, lipid material, and the least is the carbohydrate. As if you people will go into the chemical nature of cell membrane. So within the cell membrane, the maximum amount of protein around 52%. Lipid is a kind of fatty substances, is a kind of, not exactly. Okay, there are the 40%, and the carbohydrate around 5 to 8%. So that is the overall composition of cell membrane. The function of cell membrane, if you go, so it's maybe endocytosis and exocytosis. Endocytosis means anything inside the cell wall is coming, inside the cell membrane is coming, and exocytosis, anything is going to outside. Okay, so endocytosis, when the solid particles are ingested, any of the food items, if any of the organism is going to intake, like plasmodium, like protozoans, I'm telling you. Exocytosis is what? Whenever the secreted out from the cell. Okay, anything from the cell membrane is going to outside the secrete. It is called exocytosis. Endocytosis inside the cell lysis and exocytosis outside of the cell lysis. Lysis means death, guys. Okay, what about endocytosis and exocytosis? It will be much more clear during the discussion of lysosome, because lysosome also perform endocytosis and exocytosis, both type of process, and protect the cell against the foreign pathogen. Pathogen means disease causing agent. So endocytosis or exocytosis, for both two purposes, definitely every mechanism perform from the cell membrane. That's why the function of endocytosis and exocytosis is the part of cell membrane. What about the other feature of the cell membrane is the passive transport and active transport. Passive transport is what? Through the help of diffusion, okay, diffusion and facilitated transport. It's a very simple, guys. Somewhere have a more concentration, somewhere have a more concentrated material, somewhere have a less concentrated material. So from more concentrated material to less concentrated material, the particle those are going to move. It's a part of diffusions. So diffusion is possible for the solid, diffusion is possible for the liquid material, and diffusion is possible for the case of gaseous material. And see, in the case of diffusion type of process, no need of extra energy. Very simple, guys. If I am telling you I have a perfume bottle, I am going to spray this. Okay, the perfume bottle have a more concentration of the fragrance, inside the room have a less amount. So higher concentration to low concentration, directly the process called diffusion. Here no extra energy is required. Similarly, if from a one cell to another cell, any materials are going to pass across the what concentration gradients, means the concentration level both two sides is different. In that cases, movement of molecules or even ions from the higher concentration to lower concentration is going on. It is a part of passive transport. Here not even any energy is required. But if you people are going to use some energy, okay, and then finally you are going to across some material from one cell to another cell, so here through the help of energy, if you people are using and transferring the some of the substance from one side to the another side, okay, and here against the concentration gradients, against the concentration gradient means, suppose vice versa feature, suppose here have a low concentration of substances, suppose this is a high concentration of substances. So low to high concentration of substances, if you want to deliver any product, so here in that cases have need of energy or ATP. In this case, this is called the active transport. But if higher concentration to lower concentration any material is going to move, already have no energy is required, because higher to lower energy is already what happened. Here what happened? Movement of molecules are very possible without extra energy. But low concentration to high concentration, if particles are going to move across the cell membrane, here extra energy or ATP is required. ATP is the bundle of energy, you remember, guys. In the physics, it is called as a adenosine triphosphate. Now come into this next cell organelle, that is called endoplasmic reticulum. What about the endoplasmic reticulum? See, guys, actually, you know, the other cell organelles I am telling you, like endoplasmic reticulum, okay, they are generating from the nucleus and moving into the cytoplasm. Just like you see the picture of endoplasmic reticulum, it will be much more better, guys. See this picture. Here you can see the endoplasmic reticulum very well, guys. Okay, here you can see the picture of endoplasmic reticulum. See, endoplasmic reticulum is the structure. From the nucleus it is going to generate. See, it is going to generate, and this is the see, from the nucleus it is going to generate and moving out. And this is what? This is the cytoplasm as a liquid material. So nucleus to cytoplasm in between have a what? It is the endoplasmic reticulum. See, this is the endoplasmic reticulum you can see in this picture. So the location of endoplasmic reticulum, I hope everybody clear. What actually the location of endoplasmic reticulum? See, one thing more, guys, endoplasmic reticulum have a double membrane. Okay, two membranes. Just like suppose this is the endoplasmic reticulum. So one membrane is here and one more membrane is here. So that is the reason I can tell you it is a double membranous cell organelle. It is a double membranous. Double membranous, I can tell, is a double unit membrane. You remember, two layers are into the cell organelles. There are two types of endoplasmic reticulum. The one is called as a rough endoplasmic reticulum, and second is called as a smooth endoplasmic reticulum. Rough endoplasmic reticulum is popularly called as a granular endoplasmic reticulum, and a smooth endoplasmic reticulum is called as a agranular endoplasmic reticulum. Rough endoplasmic reticulum have a ribosome on their own surface, and that's why when you touch a surface it will look like rough due to presence of a ribosome. So it be a ribosome, that's why if the surface is rough, it is called as a rough. But no ribosome, if no ribosome there, the surface is smooth. But one thing I will tell you, guys, due to presence of ribosome, rough endoplasmic reticulum help in the protein synthesis. But smooth endoplasmic reticulum, it is not responsible for to help the protein synthesis. It is what? It is associated with the lipid and steroid synthesis into the cell, guys. And a part of other, guys, smooth endoplasmic reticulum is also responsible to help the cell detoxifications. Cell detoxification means, guys, any poisonous material is inside the cell, it is also going to release outside through the help of smooth or agranular endoplasmic reticulum. It means smooth or agranular endoplasmic reticulum are making a cell safe and even protect the cell through cell detoxification. Toxic substances are released outside. After it, we will go into the discussion of that Golgi complex or Golgi bodies. Golgi bodies or Golgi complex, Golgi apparatus have many names. The one as a dictyosome, the next as a Golgiosome, the next as a idiosome, or even lipochondria, different terms. One thing I will tell you, guys, in the plant cells, Golgi bodies consist of many unconnected units, and this is called as a dictyosomes. Actually, Golgi complex, you will see many of the Golgi complex are connecting to each other. If one unit of the Golgi complex are there, the single unit is present inside a plant, so then it is called as a dictyosome. So overall, I will tell you, guys, inside the plant body, Golgi complex is called as a dictyosome. Okay, so dictyosome may be a good question in your paper, where you people call the Golgi complex as a dictyosome, as a dictyosome, the answer inside the plant cell. But remember here, the Golgi complex, as it is connected many of the Golgi complex generally into the cell, but in the case of dictyosome, Golgi complex are not connected. If you go into the chemical nature of Golgi complex, the Golgi complex as a cell organelle is made up of, by protein as a maximum 75% and lipids type of material, phospholipid type of material 25 to 30%. A part of some other enzymes are also there into the Golgi complex, like galactosyl transferase, like thiamine pyrophosphatase and adenosine diphosphatase. The many of the enzymes are also there, guys. This is a property of the Golgi complex. But overall, if you go into the function of Golgi complex, so of course, in the case of the synthesis of carbohydrates, which type of carbohydrate synthesis? Golgi complex are required to cellulose and hemicellulose. And where you will see the cellulose and hemicellulose, you just have seen in the case of cell wall. Oh, it means those plant cell wall have a cellulose and hemicellulose. This one even formed with the help of Golgi complex. Absolutely right. Even those lysosome as a one of the specific and very important cell organelles form into the cell. Here in the formation of lysosome, Golgi body is also responsible. Anywhere if your objective will come, one of the cell organelle lysosome, behind it which cell organelle work, the answer is Golgi bodies. Here many people will confuse, but you please don't confuse. It is some standard question for your paper. Okay, even in the case of cell plate and the cell wall, whenever the cell is going to divide and become as a one cell to another cell. In the case of cell division, I will discuss with you. So in between, just like suppose this is the one cell, and now the cell is going to divide. So in between have a here cell plate formation, here have some cell plate formation, and finally the cell becomes separate with the help of cell plate formation, and one two cell form. This is what synthesis of cell plate and cell wall. Okay, here again what happened, guys? What happened? Golgi bodies. So Golgi bodies are responsible for a cell plate formation. That's why a single cell will separate and become as a two cell, as you people can see in this structure. After all, we will go into the discussion of plastids. Plastid is one more cell organelle you people know very well. Plastids, it is the characteristics of plant cell, and here I'll tell you characteristics of plant cell especially. Okay, plastids have a different types, especially three groups. The one group is called chromoplast. The second group you will see that is called leucoplast, and third group you will see it is called as a chloroplast. So these three different types of plastid, guys. The one plastid I'll come there, that is called chromoplast. Chromoplast is a pigmented plastid. Chroma means color, you always remember. So this is those type of plastid which provide a specific color to the plants and their own parts. They mostly contain the carotenoids. Even they have a different type of color like yellow, red, orange in colors, guys. Mostly found in the petals. Petals are actually those part into the flower. Those will have a colors. So the color of flower is actually defined on the basis of petals. You know, flower have a four structures: sepal, petal, androecium, gynoecium. You know, if you see the hibiscus plants. So hibiscus plant, I hope you can see very well, guys. In this hibiscus plant, you see. This is the hibiscus plant. So this is the sepal, guys. This is the sepal. Okay, and this is the petal. So actually the flower, the color of petal, mostly the color of petal is because of chromoplast. Even the petals, even a part of the fruit and the roots, that all the colors are formed here behind the chromoplast is there. But one thing I will tell you guys that is very, very important. Mostly people confused in the case of tomato. Okay, well, you know what is the color of tomato? Whenever it is going to red, the color of tomato is red. It is also because of carotenoid that is called lycopene. And lycopene is also a very good question. Is very important question in the many of the state pieces I have seen. Why the color of tomato is red? The answer is a lycopene. And lycopene, carotenoid lycopene is what? It is a kind of chloroplast. You always remember. Okay, you always remember, guys. And one thing more, guys. Those carrot you people are eating. Carrot is the richest source of beta carotene. Okay, and beta carotene is a derivative of vitamin A. What is the meaning of it? Very simple, guys. When you people are eating, you know, whenever you people are eating a carrot, so ultimately you people are getting a beta carotene. Inside the carrot have a beta carotene. If you are eating the carrot, so you are eating beta carotene, and this beta carotene is going to digest inside your body. In this case, liver produce some enzyme, and that's why through the help of liver enzyme, this beta carotene will convert into vitamin A. This beta carotene will convert into vitamin A. In that way, I can tell you guys, carrot become as a source of vitamin A, but not direct. Okay, firstly, carrot already have a beta carotene. Okay, when the beta carotene digests inside your body through the help of liver-based enzyme, it will generate vitamin A. Okay, but one thing I will tell you guys, those chromoplasts are derived. This chromoplast derived from the chloroplast. So what about the source of chromoplast? That the answer is chloroplast. I can tell you very well, guys. In the beginning, the color of tomato is green. You know, color of tomato is green. Why color of tomato is green? Because tomato have a chloroplast. But after ripening, the color of tomato is shifted towards a red color. It means chloroplast convert into chromoplast. I hope those statement that is mentioned in the last, chromoplasts are derived from chloroplast, that is clear, because in the beginning the color of tomato is green, but after ripening the color shifted as a red color. Now you come into the second one. The second one is called as a leucoplast. The another one is the leucoplast. What is the leucoplast? See guys, one thing, and I'll tell you, you people remember. Chloroplasts are the colorless plastid. Here no matter of the color. Okay, even not exposed to the light also. But one thing I will tell you guys, the main function of leucoplast, it is that type of plastid which store the food material. And on the basis of the various type of store of the food material, leucoplasts are of three types: amyloplast, elioplast, and alluroplast or proteinoplast. Amyloplasts are those type of leucoplasts which store the starch. Elioplasts are those type of leucoplasts which store the food in the form of fat or oils, and proteinoplasts those store the food in the form of proteins. Okay, in the form of proteins. And third one is called as a chloroplast. What is the chloroplast? Chloroplast is also one of the type of plastids and main important feature into the plant body. Okay, chloroplast is the largest cell organelle. Everybody remember. That is very, very important question. If 4 cell organelles are mentioned in your objective paper, and anywhere this question is asked, supposed, which one is the largest cell organelle? Remember, chloroplast you always take, don't be confused. Okay, and commonly found in the eukaryotes, absent in the prokaryotes generally, guys. Chloroplasts also have a double membranes, 2 membranes are there, you should be very clear, and the function is photosynthesis. That is the reason, and they are green in color, you people know very well. Plant do the photosynthesis because of chloroplast, and that is the reason plants are autotrophs and synthesize their own food material. Another cell organelle will come, that is mitochondria. Mitochondria is very, very important for all of us. Every organism, those are involving into the aerobic respirations. Aerobic respiration means in the presence of oxygen, they are generating energy. Everywhere have a mitochondria. So mitochondria are present in all aerobic organisms. Only absent in bacteria, in the blue green algae, in the prokaryotes. Neither mitochondria present everywhere, guys. The shape of mitochondria is a rod shape. Also have a 2 membranes on the outer cell layer. Okay, have it. And see, presence of DNA, Nash and Nash also discovered. So Nash and Nash even also discovered that mitochondria also have a DNA. And that is the reason I spoken you guys as in the case of 3 parents baby or mitochondrial gene therapy. If mitochondria have a DNA and within the DNA have a presence of gene. Okay, and that is the reason mitochondria also carry some of the gene that already I have discussed under the 3 parents baby or mitochondrial gene therapy concepts. But one thing more I will tell you, DNA not only the mitochondria, even chloroplast also have. A long back UPSC asked the questions. Okay, UPSC asked the question, which cell organelle is called as a semi-autonomous body? So which organelles is also called as a semi-autonomous body? Then the answer was what? Then the answer was mitochondria and chloroplast. Because a part of nucleus, guys, mitochondria and chloroplast are also those organelles, those have a DNA. Okay, in the case of nucleus, I'll show you the main DNA and genes are actually located inside the nucleus. Even mitochondria have around 60 to 70% enzymes. The largest amount of enzymes you will see in which all of the cell organelles, so the answer is mitochondria. I spoken you guys, mitochondria is a semi-autonomous body. Semi-autonomous body means cell within a cell. Okay, within a cell, mitochondria as a cell, it is available inside a living organism and genetically autonomous. Why genetically autonomous? Because they have a DNA and they have a genes. And this type of feature you will not get anywhere except the chloroplast. And this is the reason mitochondria and chloroplast both are called as a semi-autonomous body. That is also very, very important question for all of you. From the school level, you remember guys, mitochondria is called powerhouse of cell. And that's why in the form of energy as a ATP, mitochondria is responsible for energy generations. And mitochondria only involved in aerobic respiration, not in anaerobic respiration. Those all the organisms, either plants and animals, they intake oxygen and generate the energy. Behind it have a mitochondria in their own cell. Next cell organelle is a ribosome. Ribosome is available in every plant and animal. You should know guys. Ribosome is that cell organelle which is present in all plants and animals. As the largest cell organelle you people have seen chloroplast. Here I am telling you which one is the smallest cell organelle. Answer is ribosome. You people remember. Ribosome is a amembranous guys. Cell membrane is not there. This is the amembranous, that is very specific feature. Eukaryotic cell are either free in cytoplasm or attached to outer membrane of you people have seen guys. Endoplasmic reticulum have on its surface have a presence of ribosome, and that's why the endoplasmic reticulum was called as a rough endoplasmic reticulum. Rough endoplasmic reticulum means on the surface of endoplasmic reticulum have a presence of ribosomes. Okay, and on the surface of endoplasmic reticulum, those ribosomes attach. It is through the help of some glycoproteinaceous compound that is called as a ribofurans. So how on the surface of endoplasmic reticulum ribosome attach in the case of rough endoplasmic reticulum? So the answer is what? It's a ribofuran. Okay, it's a ribofuran. Okay, ribosomes are generally described as a negatively charged particles. Okay, as they have a ribonucleoproteins in this structures in the chemical compounds, but inside the cytoplasm you will see guys, ribosomes are even ribosomes, maybe many of them they are connecting to each other. If many of the ribosomes are connecting to each other, this is called as a polysome or polyribosome. In the case of the protein synthesis, this polysome and polyribosomes are working very specific into the every living organisms. Even if you go into the ribosome guys, ribosome on the basis of sedimentation coefficient, on the basis of its molecular weight, there are two types of ribosome. Even ribosome have different types, but two types are more common. The one is 70S ribosome and second is a 80S type of ribosome. 70S type of ribosome is available in prokaryote cells, but 80S type of ribosome available in eukaryote cell. Prokaryote cell means bacteria, blue green algae, mycoplasma. Okay, and one thing more guys, this 70S type of ribosome is also available under the chloroplast and mitochondria. Mostly people don't know chloroplast and mitochondria, even they also have what? 70S ribosome. Even inside a 70S ribosome have two subunits, 30S and 50S. Okay, on the basis of molecular weight, 30S and 50S size of subunits, they form 70S of ribosome on the basis of sedimentation coefficient. But sedimentation coefficient we are not going. Okay, we are not going because sedimentation coefficient we are measuring on the basis of ultracentrifugation. Okay, ultracentrifugation. Okay, everyone, you must be know guys. But the another side as one thing more, those S we people are using with the ribosome everywhere. Okay, S is the unit, and this unit is called as a Swedenborg. In one of the examination I have seen what S denote in the ribosome. So S everywhere, 70S and S all, they denote as a Swedenborg unit. And Swedenborg unit value we people identify in the molecular biology under the sedimentation coefficient. When the people are using the ribosome and their own proteinaceous structure and all, okay, then through the help of sedimentation basis, okay, we people measure this protein, and this is the reason it is called Swedenborg unit on the basis of sedimentation coefficient. Maybe in this period of time somebody somebody is not trying to even understanding, but I will tell you, you just remember S is the Svedberg unit, nothing more. You left what is the meaning of sedimentation coefficient, and in the molecular level it is required, not for general studies. Second side, you see the ATS ribosomes. ATS ribosome is in eukaryotes, also have two smaller units, as a one is a 40S and second is a 60S. When the 40S and 60S are molecular weights, they will fuse together, they form the ATS type of ribosome on the basis of, again I am telling you, on the basis of molecular biology. In the molecular biology, sedimentation coefficient and all we measure. Neither you people can think are 60 and 40, some is the 100, but no guys, it is totally different. It is totally different. Because I know mathematically 40 and 60 are same, but it is a sedimentation coefficient. It is totally different concepts. Neither 70 you can call 50 and 30 is the 80. No. It is totally different. So don't be confused. It is some of the sedimentation coefficient values. Ribosome have a ribosomal RNA as 40 to 60%. In this whole ribosome, what are the content? And 30 to 60% are the proteins, with some of the specific amino acids, arginine and lysine. So arginine and lysine are the amino acids. So which amino acids you will see in the ribosome? The answer is arginine and lysine. They are very, very important. What about the function of ribosome? Ribosome directly involved into the protein synthesis, and that's why they are called as a protein factories. You all entire bodies are basically what? Protein body. So how much the function of ribosome are doing, I hope you can understand. Even endoplasmic reticulum help in the protein synthesis because ribosomes are on the surface, and ribosome directly involved into the protein synthesis. As very simple, I can tell you. You people are near to me and I'm helping you for your selections. So I'm working as an endoplasmic reticulum. But you people are on me guys, on my surface of body. Suppose, okay, so you people are involved in your success. You people are going to involve in your examination for your success. But with the help of me, you people are going to qualify. So you people are doing the work as a ribosome, but I am working as a teacher as endoplasmic reticulum to all of you. So this is the function and features of the ribosomes. So this was the entire discussion. In this part I explained with you, and the videos or lessons are going to be very, very lengthy. So I'll stop here right now. This is my Telegram channel where you can find out the entire PDF and you people can save your time. My Unacademy profile is unacademy.com/Ravi P Agrahari 2010. Here you will find lot of the courses as a free and paid both. My code is Ravi UPSC. Don't forget, whenever you people are going to join any courses, kindly use it. It will give you extra fees discount. Why Unacademy is the best option? Because we are providing you the widest choice of educators, flexibility changes courses, unlimited views of lectures, dedicated doubt solving sessions, printed comprehensive notes, live mentorships. 178 plus toppers already got selected in 2020-21 under Unacademy guidance. Unacademy is giving you 0% EMI facilities. Here you people will get the loan for UPSC CSE. Next batch of UPSC CSE is going to start from 21st July 2020-22 in the bilingual and Hindi medium. Both here you will get the 20 GS booklets also. My courses of Science and Tech Environment are running now on the Unacademy Plus courses. If you want to join my courses, kindly use the code Ravi UPSC and connect with me. In the last, I'll advise to all of you, kindly subscribe the channel, like and share this video, and download this PDF from the Telegram. And don't forget to use this bell for the further notification and updates. So thank you everyone. I hope you will enjoyed a lot. And in the next video, again, I'll continue the series with the second part of cell biology. So thank you everyone. God bless you. Bye. Jai Hind.